Hi, everyone. Welcome to day 3 of our Pollinator Week virtual presentation series, sponsored by NIPSCO and the NiSource Charitable Foundation. Each day, we are offering enriching content that brings our new Pollinator Garden Landscaping Guide to life by connecting you to some of the experts behind the project. We're also hoping to connect you to the possibilities that await when you improve pollinator habitat at home in your garden. Maybe you're planting native species following the advice that Eric Bird and Carl Ackerman provided yesterday, or maybe after hearing from our bee experts and our Lepidoptera, butterfly and moth experts tonight, you're just going to want to go out in the garden and start to identify these incredible species. So before we go any further, I wanted to quickly review for everyone what our schedule is for the week. And now you can access this schedule through our Facebook events page, and you can also go to www.savedunes.org, click on our news tab, and the full schedule is right there for you. There's some links in here to sign up to get a digital copy of the Pollinator Guide as soon as it's released. Of course, it will live on our website once it's finalized and available. But if you want to make sure that you are one of the first to have it, then you can sign up here and we'll let you know as soon as it's available. So on Monday, we unveiled that new guide, had a lot of fun introducing our committee members and our project team members. And we're so thrilled to finally showcase all the hard work we've been putting in the past few years. Yesterday, we had Carl Ackerman of Wild Ones and Eric Bird of Shirley Hines Land Trust helping us understand how to develop that pollinator habitat in our own gardens. And today, I couldn't be more excited. We've got Desi Robertson from the Indiana Dunes National Park, who's going to be talking about native bee research that she's involved with. And we have Steve Sass and Amanda Smith from Indiana Nature, which a lot of you are probably familiar with. It's one of the most popular groups on Facebook, and it also is connected to some incredible Lepidoptera research projects on their website. So we're thrilled to have them on board to showcase their work this evening. Tomorrow, same time, same place, we're going to have pollinator art, photography, and culture programming. This is going to feature Susan Kurt, who is getting pretty famous across the region for her incredible photographs, one of which is featured here. She's going to teach us how to take pictures of that unseen world all around us. We'll have Joel Perez of the Nature Conservancy making the connections between the monarch butterfly and the East Chicago community through their Monarch Festival in September. And then we have Barb Labus, who's going to provide us some background insight into how she creates such gorgeous illustrations of native plants and pollinators, 4 of which are featured in our new guide. And then Friday, we're going to bring it home by connecting all of this rich content to the possibilities that you have to help all of our conservation partners achieve their ultimate goals of maintaining and preserving the biodiversity we have in the region by increasing connectivity to our natural areas, which are fragmented across the landscape. In between those fragmented natural areas are you and your home gardens. So on Friday, please tune in to hear from our experts. Again, Desi will be on board for that as well. So you can sign up to receive a copy of the guide on this website. Again, this information is all available on our Facebook page. And I also wanted to mention for everybody at home that if you're seeing this on Facebook tomorrow or another day, we're going to have these presentations posted on our website. So eventually you'll be able to review them and touch base on the content that was shared at your leisure. And we look forward to providing that for you. But without further ado, I'm going to stop sharing my screen and get going on our presentations for this evening. So I am so excited to welcome another friend and colleague from the region, Desi Robertson Thompson, an entomologist who's currently serving as the research coordinator for the Great Lakes Research and Education Center based at the Indiana Dunes National Park. Desi is the National Park Service lead for the Great Lakes Restoration Initiative Pollinator Task Force, What an asset we have here. Oh my goodness. And she coordinates several research projects at national parks throughout the Great Lakes region. These projects focus on understanding how best to conserve our pollinators, especially native bees, how restoration efforts can best benefit them, and how pollinator communities have changed over time. So thank you for being with us tonight, Desi, and you are welcome to share your screen and take it away.
Great. Well, thank you so much for that introduction. Let me see, let me get this going here. Make sure I can, it works so easy and when we practiced it, huh? Let's see here. Go here, share. Is that working for everyone?
Perfect.
Okay, great. Well, thank you all thank you, Victoria, for inviting me. Thank you to the whole crew here who has just done an amazing job on this landscape guide. I'm so excited that to be a part of it, and I'm so excited for everyone in the region to be able to see it. I think it's going to inspire similar efforts across all over the place, like across the country, probably. It's just really, really exciting. So today, I wanted to just mostly show you all a bunch of pretty pictures of bees so you can all fall in love with them the way I did and want to help conserve them. But I am going to talk about some of the projects that we're doing at Indiana Dunes National Park to understand their needs a little bit, so how we can best help them. I'm going to start with giving you a really quick background on native bees, and then I'm going to dive into some of the research. And if there's time, I'm going to finish off with a couple of ways that you can add habitat that will specifically benefit native bees. So in terms of pollinators, there are all different kinds of pollinators. Many folks are familiar with the monarch butterfly, which is a member of the Lepidoptera. I think, Victoria, you might have first said that this is sort of the gateway drug or gateway bug to other pollinators. And certainly Lepidoptera are very important pollinators, especially moths. So I'm really excited to hear from Steve and Amanda to hear about their work with moths. Some other important pollinators, of course, though, are wasps. Many adult predatory wasps will feed on nectar, so they can aid in pollination services. Lots of flies. So while fly larvae tend to feed more on detritus or dung and things like that, the adults oftentimes also nectar. And so they can be really important pollinators as well. And also bees. There are some bee species that are incredibly important pollinators. But what I want to talk to you all today about are, of course, bees. And bees are some of our most important pollinators, and there's three main reasons for that. One is they are the ones that are actively collecting and transporting pollen. Okay, so bees will collect pollen. They make a pollen ball. Sometimes this is called bee bread. And they bring that, the females bring that back and they lay an egg on top of that in their nest. And that's going to provide nourishment for their developing larvae. So they'll eat that until the larvae will pupate and become adults themselves. So bees are really some of the only insects that are actively collecting that pollen. They also do something called flower constancy. And what that means is that when a bee is foraging for pollen or even for nectar, they tend to stick to the same species when they're traveling from flower to flower. And that really helps to ensure pollination of that flower species. So for instance, one foraging trip, a bee might fly from apple blossom to apple blossom to apple blossom. And on the next trip, maybe they'll go to dandelion to dandelion to dandelion. But generally, and there are exceptions and it's not totally consistent, but generally they're not going to go apple blossom to dandelion, apple blossom to dandelion. It's really more efficient for them to kind of stick to the same species. So the third reason that bees are the most important pollinators is because they regularly forage in areas around the nest. Now, this can be great news for farmers and gardeners because if you can provide nesting habitat to encourage native bees, then, and you keep those near your crops, that will really help provide those additional pollination services where they are needed the most. Now, when I'm talking about bees, I'm not talking about the exotic European honeybee, which is actually not a native species to North America. I'm talking about native bees, and there are about 3,600 North American bee species. And again, I wish I could talk about all these beautiful bees. I encourage you to get the landscape guide when it comes out. And we have a quick overview of some of the main groups of bees, talking a little bit about their biology. But you can see, I mean, so many different kinds of bees. And just to give you an idea of the regional diversity is in Wisconsin, 388 species of native bees, in Michigan, 465, neighboring Illinois, 400. And here in Indiana, we have approximately 416 species. Now, that's a lot. That's a lot of bees in one state. But what's even more amazing is that half of that species diversity occurs in the narrow strip of area in the region known as the Indiana Dunes. And the reason for that is because of the unique diversity of habitats we have, the diversity of plants, and also the diversity of soil types, and even diversity in climates, microclimates. You know, the climate on the south face of a dune is very different than in a humid forest, maybe in the Valparaiso moraine. So that's why we probably have a lot of diversity here of bees. And I just want to point out that we know that we have all this diversity thanks to the incredible work of the USGS researchers who are collaborating with us on this project, and some of these projects that I'm going to talk about. And here they are. So before I move on to those projects, I want to acknowledge my collaborators, the bee team, especially Noel Pavlovic and Ralph Grandel of the USGS, who are in many ways the brains behind these projects. Noel and Ralph did some of the original foundational research about 20 years ago and really set the stage for a lot of the research that we're doing now. I also want to point out, give a special shout out to Johanna Nufosi with the USGS and Tyler McGill with NPS, and who have been doing most of the field work to actually accomplish a lot of this work. Johanna also does much of the analyses for this project, and Tyler has become our regional bee expert. He processes all of our bee specimens and identifies many of them. So again, as I mentioned, I wish I could just spend this whole my whole time just talking about bees and showing pictures, but really I'm going to talk about why we are interested in bees. And the main question that we're looking to ask in our research is, how can we best manage for the conservation of native bees at the park? What can we do, given this incredible diversity that we know of, and understanding that there is a decline of global pollinators? We want to better understand what is happening here in our region, in the park. And when we look at Indiana Dunes National Park, we know that it has, as Victoria pointed out, it's a mosaic of unique, high quality, rare habitats across the park, but they're highly fragmented, right? So these high quality habitats are interspersed with some degraded habitat, areas where we have invasive species, heavy industry in many cases, housing developed, agriculture. There's a lot going on here. And so, you know, I just want to mention when I talk about high quality habitat, specifically for pollinators, I'm talking about something like this in these images here. So here you can see some sites. This is in Miller Woods, and you can see that it has a fairly open canopy. There's some black oak trees, and it allows, you know, there's openings in the canopy, so there's a lot of light that can hit the ground, and that supports this abundance of floral resources in the understory. And that provides, that's an important resource for pollinators. And the reason this is high quality habitat is because this has been restored or actively managed through the efforts of the Park Service and partners. So through prescribed burnings, through removing invasive species and that sort of work. Now, if you contrast that to low quality habitat, it was an example here, This is an area that has been really encroached in. You can see, it's hard to maybe see these pictures here, but this has got a lot of different invasive species. I see some Asiatic bittersweet in there. It's being encroached by woody shrubs and trees and some other invasive species. And that crowds out much of that understory herbaceous plant that is so, floral resources that is so needed to help our pollinator species. So if you think about a bee, you know what that does to a bees. Imagine you've got these sort of two islands of high quality habitat, and I know Eric Bird touched on this a little bit yesterday in his presentation. But if you have these areas of high quality habitat, but they're isolated, that can be a barrier to individuals moving across and species moving. That can have a negative effect on the population through reduced genetic diversity, and just it's not good. And so you know, they can't move across, and they're separated. These are not happy bees. But if you can restore these areas, if you can connect these islands, create a bridge or a corridor, and so they're no longer isolated, you can begin, at least our hypothesis is that by restoring these areas, we can increase the connectivity and we'll begin to see a positive response in the native bee community. So we'll see increased diversity, we'll see larger numbers of individual species, and so on. So that is one of our primary research questions. What are the effects of our pollinator habitat restoration efforts, specifically in oak savanna? We want to know, how will the native bee community respond to increased connectivity across this oak savanna landscape? Are we going to indeed, you know, see a positive response? Whoops, I went the wrong way here. There we go. So as is the case, we have some great historical data thanks to the work that Noel Pavlovic and Ralph Grindel did previously. We have some good baseline data. We also have a lot of information about how these areas were managed. We know what actions have taken place in terms of prescribed burn, invasive species removal, if we've enhanced any areas with native seed installations or plugs, and so on. And so we can use that information to kind of give us an idea of how the area has changed over time. But also, while we are going through and actually restoring these areas and working on, so through Great Lakes Restoration Initiative funding, we are doing a lot of restoration of some of these areas. We can continue to monitor these areas for changes. So we can monitor the pollinator community as it changes, and we can evaluate the success of those restoration actions. So just as an example of some of these on the ground habitat restoration and enhancement activities that we have, we're focusing a lot of our efforts along this Marquette Trail corridor. And so this corridor is kind of this narrow, if you've ever been to this area, and I really encourage you all, it's a great trail to get out on, and even more beautiful since they've been restoring it and since bringing back some of these floral resources. It's just wonderful. And by removing these invasive species, getting rid of some of this shrubby underlayer, we're beginning to see a change. You can see now we have better floral resources. And so we can look at the pollinators now. We're continuing to collect pollinators, looking at their community and say, hey, is it actually working? Are we seeing a difference? And just to give you a little understanding of what that means when we're doing this research, is we set up transects in different areas. And so some of these are the same transects that where data were collected over 20 years ago. So we have revisited those, and we look at it immediately before the restoration actions, during the restoration actions, and then in the future, we're going to continue to look at these and see how it changes. But we set up these transects, and we use these little bowls filled with soapy water, and that's how we collect bees. And for a lot of bee species, unfortunately, in order to identify them, you need to look at them under a microscope. And so we do, for this project, we do end up collecting some bees. So we don't have any results in all these studies that I'm presenting today. It's going to be sort of like, stay tuned in another couple years. We'll be able to tell you what's happening. But this is one project I wanted to mention. A larger project that we are doing is also focused on this idea of habitat connectivity. So while we have restoration activities going on in some specific areas, and we can see what's going on with those and try to understand them, we also want to look at how we might target our future restoration efforts. We want to be able to identify specific habitats or sites that may need to be prioritized or targeted for our restoration activities. So, the idea is, if we can restore these small little degraded areas, maybe that connect to higher quality sites, if we can identify those areas, then that maybe gives us more bang for our buck in terms of restoration efforts. We always want to restore as much as we can, but if we can do it in the most efficient, effective manner, that obviously is even better. So to do this, we have this large project here, and I don't know if you can see on this map across all these little green dots, that each one of those dots represents one of our sampling sites. And we quickly realized in this project that it would take far too long to follow the same sampling protocol that we had been doing along that Marquette corridor, because it takes, might take an hour to get to each of those sites. They're far apart from each other. You got to hike in. Sometimes you're climbing, and I only do this every once in a while. So this is really for Johanna and Tyler, who are doing the real heavy lifting on this project. Climbing through raspberry patches, stinging nettle, and to access these far off places can be really challenging. So we had to think about how to modify that research design, and we decided to focus our efforts on cavity nesting bees. So cavity nesting bees, they use hollow tubes or holes in logs for nests. So the female, she will lay her eggs inside of these holes, make these little individual brood cells, and provision that with pollen, and then make another brood cell, and that becomes the nest. So we thought, okay, if we can provide a standardized sort of nesting setup across at all these parts, we can start looking at occupancy levels there. So we built several hundred of these bee blocks that you can see right here, this picture right here. They have they're designed to have 4 different sizes. So some species of bees like the big sizes, some like the little tiny sizes. So we can see what species of bees come in based on that. And we also have them set at different heights. And with this, we can go back and we can monitor these from time to time and see if they are plugged. So in this picture right here on the right, this looks like it has been plugged probably by a leafcutter bee. And leafcutter bees just like the name sounds, they cut little pieces of leaves, and they will use that to line their nests and also to plug the holes in their nests. So this is an example of one that has been plugged. And so this is, if you come across these, if you're in the park hiking and you see this funky setup, we use tomato cages to mount them, because that was our most efficient, both in terms of cost and quick deployment method. So, and you see all these weird little blocks. That is part of our research project, and we ask that you observe it from afar and know what it's about. So what we are doing now is we do a couple different rounds of sampling these. We sample. We look for occupancy plugs. We also will do some traditional bee bowl sampling. We collect all kinds of habitat data, all sorts of things. And eventually, we can plug all this into sort of the computer, so to speak. And this is really where USGS is the brains behind a lot of this project, and looking at how we can sort of look at different levels of what we call high connectivity across the landscape. So we have hypotheses of areas that we think might be important in terms of connectivity, and we can kind of change different features of that to make it higher or lower connectivity. And then we can look at the data and see what the data have told us, if in fact this is important. Are there specific areas in all of our sampling points all across the park that we really want to target for restoration efforts? And what are some of the areas that have really high diversity? What are those like? What features are we seeing there? And do we want to try to replicate those in other areas? What are the things that might be missing at a particular site? What do the bees need? So I just want to finish up with a quick little segue into sort of what you can do. Obviously, we're trying to do as much as we can at the national park. The land trusts are all of our partners. NIPSCO right away. We have really large tracts of land, and that's great. We can try to do as much as we can. But as Victoria said, there are also places you can continue to add to this connectivity. You can help bring those bees together from those high quality areas, from the national park to maybe a Shirley Hines property. And there's lots of houses, lots of neighborhoods in between there. And if you want to help bees, specifically, some of the things that you want to do are provide some year-round floral resources. Bees need more than just flowers in June and July, when a lot of folks like to have their pollinator gardens. So there's some bees that are early emerging species that are kind of forest specialists, for instance, and they need flowers at the end of March, early April. And so if you can have native species that bloom around that time, that's really, really helpful. If you can, similarly at the other end, if you can provide species that bloom in the fall, things like New England aster and some of the goldenrods, those are really important species that help bees kind of get ready for winter and provision resources. You want to have a diversity of a diversity of flower shapes. Different species tend to be like specific flowers in different species. There are a lot of specialists. And so if you have a diversity of flower shapes, you should be able to support a diversity of bee species. Now, if you remember, I mentioned how bees exhibit that flower constancy, where they like to visit the same species over and over again. You can help their efficiency even more if you plant species in the same, if you plant them in groups together in masses. So it's nice to have them all kind of mixed up, and I do enjoy the way that looks too, but maybe have some areas where you have, okay, here's where I'm going to have all of, I don't know, I can't list any plant species off the top of my head right now, of course. I'm not a botanist. But here's where I have maybe all of my butterfly milkweed. And maybe here's where I have all of my hairy puccoon. And maybe here's where I have all of my lupin. And that orders a little bit, but it also helps those bees be able to get to those areas a little bit more efficiently. Another thing, so not just on the large landscape where you want to increase connectivity, but even in a small yard. So maybe you've got a great pollinator garden and it's in the middle of the yard and it's surrounded by lawn all around that. When a bee flies from, maybe a forested area or another garden across that lawn, it's like a landmine for that bee. It becomes a target for birds and other predators. So if you can provide sort of like a little cover, maybe it's just a little trail of cover for the bees to get to, that will also help. Use native species, avoid pesticides as best as you can. And finally, I want to mention nesting resources, which I have a quick little image here I'll just show you. Bees nest, as you know, in all different sorts of places. They nest in dead stumps, branches, trees. They might nest in old stems. A lot of species also nest in the bare soil. So if you can provide all these things on your lawn, you are going to help the bees. And with that, I'm going to leave you with my contact information, and also this fun little GIF I found from the University of Minnesota of a bumblebee doing buzz pollination. And you can see how when they vibrate and do their sort of sonification and buzz, the pollen just pours out over them. And I just thought it was a really cool image that I wanted to share with you all. So that is all I have. Thank you.
Wow, thank you, Desi. That was so fascinating. And to finish off with that video, I can't believe it. It's incredible to think. I mean, the bumblebees are those large bees that we often see flying around on the tiny little wings, but it was through working with you that I learned about buzz pollination, and to see it in action is extraordinary. So I wanted to check in with Katie. Do we have any questions in our Facebook
We do
chat?
have, we do. We have a question from Jim. So he asked, how long would it take a species to move through a newly restored corridor?
That's a really great question. So there have been some studies that have shown that it's really quick. And just within a couple, bees, bees can, are active flyers. They can fly, and, they're mostly limited by the distance from their nesting site. But in terms of dispersing, and, there's sort of the distance that they fly back and forth to forage for food and come back to their nest. But then there's also their dispersal distance, and some species disperse further than others. But so there's a little bit of limitation there. But some of the studies have shown, within a few short years, that if you have a fully restored, area, they'll find those resources pretty quickly. And so get started as soon as possible, get those pollinator gardens started as soon as possible, and you should start seeing bees showing up.
Yeah, that's great advice, Desi. I've got some cut stumps in my backyard, kind of tucked into a corner, and I think I'm just going to go out and drill some holes in there and try to attract my native bees. Those mason bees are beyond adorable. I need to see more mason bees. So thank you, Desi. And if any more questions come up for for Desi, please do put them in the Facebook chat, and we'll try to get to those at the end of the presentation. Or please do reach out to Desi or to Katie or I, so that we can put you in contact, and stay tuned for the results of her research. I can't wait. I can't wait.
I have a quick question for Desi, if I may.
Sure.
The tomato cage houses that you put up, what are you finding as the ideal height? Are some more being more populated than others? And you purposefully use different heights of boxes, correct?
Yes, so we have some that we have two sets. So we have one down below, and I'd have to look up, I'd have to ask Tyler, my biotech, exactly what the heights are, but I think one's at like 18 inches, another one might be at 2 feet or something like that. So it's on the lower rung of the tomato cage, and then a higher, the high rung. And I think, we're really just they just finished the first round today, in fact, or actually tomorrow, I think, is they're finally finishing. But what seems to be happening is they are using the lower rungs. From what I've seen, it looks like they are using a lot of the lower ones. But the funny thing is, now later in the season, the vegetation gets higher. And so we might see a difference seasonally, where once the vegetation kind of gets high and covers it up, then maybe they can't find those as much, and then they might occupy the upper ones. So don't have a very good answer for you yet, but that might be what we see.
The reason that I asked is I see these for sale, these bee houses for sale quite commonly now at big box stores and even supermarkets, and
Right.
they don't seem to come with instructions like, to whether or not to hang them at 6 feet up in the air or a foot off the ground.
Right. And, the jury's still out in terms of how beneficial those bee houses are in terms of helping native bees. I think they're an incredible educational resource. But one thing that if you look in the Xerces Society, they have some great resources in how to create these and kind of how to manage them. Because one of the issues with some of those bee houses are that bees that nest there may be more prone to predation. They may be more prone to pathogens, and because it might be an unnatural sort of aggregation of bringing them together. And so there's some information out there on how to clean them, how to, disinfect them from year to year, and to make sure that you are not adding to the stress of native bees.
Wow. Wow. Really great question, Steve, and really insightful answers, Desi. Thank you. So much food for thought. Wow. No, I'm thinking I was going to get a bee house. Now I'm like, I don't think I'm going to get it. Anyway. That was
A few dead logs worked just great.
Yes. Yes, that was wonderful. And we can't wait to hear more, Desi. Thank you again. We're going to transition now into the second half of our presentation series this evening, and are beyond delighted to welcome Steve Sass and Amanda Smith to the stage. They are the co-founders of Indiana Nature LLC, which includes the educational initiative, indiananature.net. If you haven't checked that out, I encourage you to do so. It also has a corresponding social media outreach page, as well as the Ecometric brand. Their popular Facebook group In Nature, which I check every day and love, contains the Great American Indiana Nature or GAIN projects, which have engaged thousands of Hoosiers to collaboratively build a database of moth, butterfly, tree, and wildflower populations in the state. Talk about citizen science in action. Steve and Amanda have collectively served in numerous capacities for Indiana environmental organizations, including land trusts and Audubon societies, native plant societies, for several decades. A little about each of them individually. Amanda resides in Hamilton County, where she is the superintendent of natural resources and education for Hamilton County Parks. Steve is a small business owner in South Bend. One of their most recent collaborations has been co-authoring a new natural resources management plan for the city of South Bend, venues of Parks and Arts Department. Huge success. So welcome to you both. And we're so excited for your presentation. Please take it away.
Thanks for having us.
Going to attempt the screen share here. How's that working?
It's good.
Okay, so thank you so much. We are incredibly honored to be here and be a part of this, the second volume of the Living in the Dunes Guide for such a wonderful, prestigious organization as Save the Dunes. So we're here tonight to talk about Lepidoptera specifically, and we're going to focus in on their role as pollinators, and we'll talk about some other interesting aspects to them as well. Since you already just read our biography, we probably don't need to go through this again, because it was actually much more comprehensive than than the slide that we put together. But essentially, I live in St. Joseph County. I grew up in Michigan City in the Dunes area. Amanda is from Indianapolis originally, and she is currently residing in Hamilton County as the superintendent of
All that stuff. We don't have to go into it. We have more fun things to talk
We
about.
already said that. Okay,
Yeah.
so a little bit about Lepidoptera. So first of all, what are Lepidoptera? Lepidoptera are an order of insects, one of many orders of insects that consist of the butterflies and the moths. And so these are some examples of what other orders of insects would be, including Hymenoptera, which Desi just talked about, which includes all of the bees as well as ants and sawflies. So we're focusing specifically on the butterflies and moths, or the order of Lepidoptera. The order of Lepidoptera itself is fairly extensive, and it's divided up into superfamilies. And so if we look at the superfamily we have on the top right of the screen, Papilionoidea, which is all of the butterflies and skippers. So every butterfly and skipper in the world are categorized into that one superfamily. Contrasting with that, the animation shows all of the families of moths in the world, superfamilies of moths in the world. So there are 40 some superfamilies of moths for just the one superfamily of butterflies. So they're an incredibly diverse group of insects, which are not probably noticed as much as the butterflies are, because they're not as brightly colored, and they're not flying. Most of them are not flying in the day like butterflies are. So we'll touch a little bit about what the differences are between butterflies, moths, and skippers. On the left image, we have a giant swallowtail butterfly. Butterflies typically have relatively small bodies to large wings, versus moths and skippers are typically the opposite, where most of them have somewhat large bodies to smaller wings. And so we have a moth in the center, a common pug, and a skipper over on the right hand side, which is the least skipper, which is actually the smallest butterfly, or the smallest skipper, which is part of that super family that contains butterflies, the smallest one we have in Indiana. Another difference is the antennae. So if we look at the antennae on butterflies and on skippers, they have a club at the end of them. Moths never have clubbed antennae. Moths have various different types of antennae. We're going to look at a few of them coming up in some slides, but they're never clubbed. Also, butterflies and skippers most of them, or just about all of them, I guess, would be diurnal. That is, they're flying and active during the day, versus moths are typically nocturnal. Not all of them, but the majority of them are more active at night than they are during the day, which is probably one of the reasons why we don't recognize as many of them as we do our butterflies and skippers. So we'll touch on the life cycle of butterflies and moths, and there's a really nice graphic in the upcoming pollinator guide, Save the Dunes pollinator guide, that talks about this as well. Complete metamorphosis. So this is the life cycle that Lepidoptera go through, and we could jump in at any point in the life cycle, but we're going to start, for the purposes of this illustration, we're going to start with the eggs. The eggs hatch into caterpillars, and it's important to note that the female Lepidoptera will deposit the eggs, her eggs, or oviposit, onto or near the larval host plant. And by larval host plant, we mean the food source that the hatched caterpillar is able to eat. And from that point, the caterpillars will go through several different growth forms or growth stages called instars. And so in this photo, we have a early instar caterpillar of an imperial moth. And by the time it's done going through 5 instars, Amanda, how much does it actually grow? You had
So proportionally, it grows typically, depending on the species, but about 2,000 times from the size that it came out of the egg. So that would be the equivalent proportionally of an infant growing to be the size of a school bus in 2 weeks for these guys generally.
So this is really the primary feeding stage of this insect's life cycle, where they eat and eat and eat. We've all heard the story of the hungry caterpillar, and that's not a misnomer. They do eat a lot, and they need to have specialized food. We need to really stress that more than anything. Having the right plants in our landscape is what is allowing these the life cycles of these butterflies and moths to continue. So from there, once they reach a certain size, they pupate. They go into, in this case, this is a silk moth that builds a cocoon where a miracle happens. And a while later emerges the adult moth. And this is an imperial moth, which is a very striking and beautiful moth that's native to our area. And then that moth will mate and hopefully mate and lay eggs, and the life cycle will, if everything is successful, it begins again. So host plants, again, are extremely important, and Victoria uses the term the gateway bug to describe the monarch butterfly, and that's very much the case in my experience, our experience as well, is that just about everybody knows the story of the monarch butterfly's relationship with milkweed being its host plant, or the only thing that its larvae is capable of eating. Lepidoptera are really all considered specialized eaters, and from that, I'd like to compare versus people. We can store and we can buy there's hundreds of different things that we can eat there. For Lepidoptera, that's just simply not the case. And they have evolved very specialized dietary needs. So in this slide, we have a black cherry tree, a Prunus serotina, which contains chemicals that will turn, when eaten by a mammal, will turn into hydrogen cyanide. And so it's poisonous to people and to other mammals. but it is one of the host plants for this beautiful butterfly, the eastern tiger swallowtail, which incidentally, you can see the image of the caterpillar below the person. This is a bird dropping mimic as a caterpillar. So that's one of its life survival techniques. But host plants are very important. And they have different degrees of specialization in Lepidoptera. This is a slide we put together on for the Bombicoidea, which are the silk moth superfamily of moths. At the left-hand side, we have the what we're calling the specialists, such as the Cecropia moth, which can eat from plants of 74 different genera in Indiana. And as we travel along this slide over to the right, we become more and more specialized by some of these insects, where some of them over on the far right are only able to eat from 1, 2, or maybe 3 different sources of food. So, the downside of extreme specialization is that if something were to happen to their food supply, the Carner blue is a good example, where it can only eat from one species of plant in the Indiana Dunes area. And if something were to happen to that population, it just simply can't move on to another plant. It has to have that larval food source. So, how do they find the host plants, Amanda?
Well, I was curious about that one day, just watching my milkweed and waiting for a monarch to show up. So as most of my curiosity leads me, I asked the question and found the answer. Essentially, our moths and butterflies have amazing sense of smell through the olfactory, these specific cells, primarily on their antenna, that lead them to the plants that they're programmed or have evolved to use as a host plant. So the plants give off a VOC. If you've ever painted at home, you've looked for paint maybe that has a low VOC, volatile organic compounds. When we smell cut grass, that's a VOC. So these plants are kind of emitting in very small percentages these types of compounds that the butterflies and moths are looking for. So in the case of milkweed with the monarch, it's very specific to that genre of plant. If we go to the next slide, we can see this is a Virginia creeper sphinx moth, and they only feed from the grape family. And so they, this is a wonderful, very good photo of one. And those kind of olfactory receptors that they have are on the antenna, but they're also on the legs. Butterflies will smell and actually taste with their legs. So if you watch a moth or butterfly, they tend to kind of move their feet around. They're actually tasting the plant source that they're on. And then in some cases, moths and butterflies will have palps. They'll have the little kind of mouth parts, and those also have those receptors on them. So our insects that they definitely, both of these, all of the Lepidoptera, well, the order of Lepidoptera are pollinators, but not all of them pollinate. So today we're going to focus primarily on just the species that do pollinate. In Indiana, we have about 11,000 species of moths. So lots of moths. And there's not been great studies on this moth pollination, but in Europe, they have done more studies, and they found that about 45% of moths were pollen carriers. So hard to say for sure what percentage ours are.
And you mean 1,100, I think, right?
I'm sorry, not in Indiana. That was for the US.
Oh, okay, in the US.
Sorry. Yeah, thank you. And we can go to the next slide. So pollination syndromes, we're not spending too much time on this, but I found a really interesting story. Our insects, though, have co-evolved with our flowers, and angiosperms in particular have utilized insects to transfer pollen in order to create more flowers and more plants. And there's pollination syndromes. If you go and look back, you can look and see that certain butterflies have kind of vaguely things that they're looking for, and then moths for certain things, bees for certain things. Pollination syndrome, though, was actually kind of brought forth through, of course, Darwin, who had been sent a flower from the island of Madagascar from a colleague. And the flower itself was an orchid, and it had a very long tube, and the nectar was positioned down at this long long tube. And he actually was quoted in saying, "Good," he said, "good heavens, what can suck it?" Asking another colleague in writing, what on earth, what insect has a long enough pollination device, a proboscis in this case, that could reach 11 inches down to the nectar source. And he hypothesized that it had to be a moth based on the pollination kind of um, specialties. Typically, white flowers, especially and many orchids, will have moth pollinators that have co-evolved. So he hypothesized that it was most likely a moth with a proboscis that would reach between 10 and 11 inches. 20 years after his death, that moth was actually finally found in the island of Madagascar and was named the Morgan Sphinx moth for the Darwin's orchid. So kind of fun to look back on that. Pollination syndromes are not concrete, and we can go to the next slide to look at some of those. But these insects are looking for a nectar reward in some cases. Some of them will eat pollen or feed on pollen, but for the most part, our Lepidoptera are going for nectar. Nectar is really just a byproduct the plant is sort of making for these insects in order for them to get a reward for pollination. It's essentially just a sweet liquid, depending on the flower, the species, and also the time, how long it's been open. It may be 8 to sometimes 50%, sort of the turquoise of the sugar compound, but also has other vitamins and amino acids to it. So generally, butterflies like to go after maybe red or different kind of bright flowers. Moths, white, they tend to like the pink. They don't necessarily need super sweet smelling flowers to be attracted to them. And a lot of like Jimson weed and a lot of the white blooming flowers are attracted by moths at night. And Lepidoptera can see in the ultraviolet light. And this was actually a photo I recently took of a St. John's wort growing here locally in central Indiana. And I have a black flashlight that I am going to use to find caterpillars this year. And I decided to use it on the flowers. So it just gives you some idea of the differences to our naked eye to how a Lepidoptera would see. They don't have very sharp vision, but they're definitely seeing in different spectrums. We have one tribe of moths that are well known as pollinators and has actually been studied the most. And this includes some of our more favorite ones and also some of our least favorite ones, including the Carolina Sphinx, which is on the left. This species is the tobacco hornworm, which is what does eat our tomato plants. I'm kind of against calling them worms. I'd rather them call them caterpillars, because I think it's a lot easier for us to be grossed out and not share our plants with something called a worm than it is a caterpillar.
And so, incidentally, the name of the subfamily Macroglossini is Greek or Latin or combination, meaning a large tongued.
And I think it's the Carolina Sphinx that has one of the longest tongues in our state, at least. This is, though, more of a settling moth. So these moths have not been well studied. And this is a common moth. It's also called the common looper, and it's a rester. It settles on just sort of the discs of plants and the flowers of plants. This moth actually is so well known, especially for orchids, that it carries pollen. And these settling moths, unlike the sphinx moths that have the long proboscis, are probably better pollinators because their bodies are furry and they're just resting right on the plant. And in this case, for the common looper, it's not uncommon for them to get pollen in their eyeballs, that sticky pollen right in their eyeballs. So this picture just shows what, on the common milkweed, what just a bunch of moths would look like. So these are effective pollinators, but probably aren't getting near as much credit for pollination in regards to what they're actually doing. And that's just based on limited studies.
And so not all moths are pollinators. In fact, probably most of them, the vast majority of them, are not pollinators. The silver moths in particular, though, we can't not talk about because they're so beautiful and so interesting. This is the largest moth in North America, Eastern North America, the Cecropia moth. And it's one that if we if we look carefully, we might find in our own neighborhoods. The silk moths are very interesting because not only do they not pollinate, they're not they're born, or the adults are emerge without a digestive system. There's no mouth parts, there's no digestive system at all. So they live long enough only to hopefully find a mate and reproduce. And so they're, as adults, they only last just as long as the energy that they're transformed with or metamorphosized with. But all of the moths and the butterflies are part of the food web, and this makes them very, very valuable. It's probably the most valuable um, asset that they have to the ecology and to our food web. And right now we have all of these birds that you see in this photo are nesting in Indiana, in the Indiana dunes. We have a yellow warbler, a common yellowthroat, a chipping sparrow, and a song sparrow. And in their mouths are insects, and I think in most of their mouths are caterpillars. Caterpillars are soft-bodied and they're full of protein, and they are the number one food that these birds are using to raise their young. We put out bird feeders full of sunflower seeds, but these birds can't use sunflowers to raise their baby birds. Baby birds can't eat sunflower. They need to have protein, and the majority of the protein comes in these soft-bodied insects. The chickadee was one that was part of a study by Doug Tallamy, who wrote Bringing Nature Home, of course, and through his research project, they determined that a single clutch of chickadees requires about 8,000 of these caterpillars to be able to raise. And so these birds are working very, very hard day in and day out, flying into the trees to find caterpillars to bring back to their nests. And so it's really important that we maintain a healthy amount of caterpillars in our ecosystem because the birds are depending on it. You can
we can and have done an hour-long program almost completely on how these moths and butterflies avoid predation. They really don't want to be a lunch for whether it's a caterpillar or a bird or a bat, or there are a lot of things that eat these guys or prey on them as parasites. So and there's just amazing defense and strategies and adaptations that they have to avoid predation. We're going to focus on one, which is sort of well known. The swallowtails have that long, beautiful little kind of tail on both sides of their hind wing. The Luna moth has something similar. And these two are, these are an example of how they have adapted essentially to avoid predation. They have vastly different types of predators. Both are flying predators, but the swallowtails are generally pollinating and going about their work as butterflies during the day. So birds are their primary source of predation and impact. For Luna moths, it's bats. They're flying at night. So for the Luna moth echolocation, these bats are using echolocation, and the tails actually will hopefully flutter enough in a way that will attract the bat to the tail. And hopefully the bat will hit the tail and maybe pull off a large section of the tail, which you can see, which happened on the left-hand photo for the Luna moth, but saves the body. The body is obviously what they can't really lose. And you've may seen butterflies and moths outside that are pretty beat up. And the wings can take some abuse and still be able to fly. The swallowtail on the right-hand side has actually lost both of its tails. So most likely was preyed upon by a bird. The bird missed the insect and it lived to fly another day, hopefully.
So we started the Facebook group we talked about earlier, or Victoria talked about earlier, back, what has it been, 5 years now? And 4 years, 5 years, something like that.
It's 5.
5 years, yes. And we were getting all of these amazing photos from around Indiana. And we realized that, social media is very fleeting, and the photos are here today, gone tomorrow. And we decided that we wanted to come up with a way that we could involve people from around the state of Indiana to begin compiling a essentially a database of photos, and along with dates and times of different Lepidoptera. Amanda and I both had an interest in Lepidoptera coming in, and we thought that would be a fun project to do. And so we started doing that, and gosh, what, 2018 was the first
Mhm.
year we launched this project called the Great American Indiana Nature Lepidoptera Project. And as these photos began being posted to the Facebook group, we then started a database. And this is a screenshot of the database. You notice the numbers over on the right-hand column. Those are the actual numbers that we have in the database. I don't know if this is the very bottom of the database. It's probably pretty close, but we're over 37,000 individual sightings now, right?
Yes. And as of today, we have cataloged it's 1,784. We had a new gold star moth today, which I actually have a slide of below, of different species. So 1,784 species of Lepidoptera through the GAIN LP. And we have probably 160 participants in it. And so of that total amount of Lepidoptera, only about 115 of them are butterflies, the rest are moths. So about a 1-to-10 ratio, which is matches what it is for really worldwide. There's about, for every one species of butterfly, you have about 10 species of moth.
And so from that data, we began building species pages on our website. And we started with the silk moths that we talked about earlier. This is an example of one of our web pages, some of the content, which includes taxonomy, the etymology, where the name came from, how to pronounce it, various featured photos, the North American range that we based on other websites, active data. The conservation status. Then we go into our database is directly tied to a map of Indiana, whereas if a sighting comes in from a particular county, that county will change immediately from white to green to indicate that species has been found in that county. On the right.
Interesting data coming from the counties. We're seeing some interesting data, even though we're still young and kind of collecting, but lots of moths that are having a more kind of southern trajectory. The regal moth is one that I remember here in central Indiana before I was really interested in moths about 10 years ago, and we're not seeing it now. So some interesting changes I've noticed just in my dealings.
The web pages contain a table with the known larval food sources that we have in the state. And so you can look at this if you're trying to attract a particular moth. You can see if you have the host plant for it. And then some of the data we've been able to extrapolate from the sightings. The bar chart at the bottom is one that shows the number of sightings per month by the life form, and so the green are the adults. So this is the Luna moth, I believe, correct?
It is, and I picked the Luna moth data because I thought it pretty well shows the two broods that we have of Lunas in the state. So we have an early brood, and then we have a second brood, and so that data is panning out based on what we're getting submitted.
At the bottom of our species pages, we have the photo gallery of all of the photos that were submitted to us either by the Facebook group or through email. And then this year we started adding these phenophase codes to them as well to indicate whether or not what life cycle the photograph was. And we added we have an interesting one, which is flower visitation, the FV symbol on there. And this is one that doesn't visit flowers, but this flower visitation would be, for example, if we have a picture of a butterfly pollinating something, it would be classified in our database as a FV. And then we can go back later and we can pull up all of the FVs for a particular species. And then we can look and see what flowers these particular species are pollinating. And we've had some interesting things come from that, right?
Mm-hmm. Yes, we've definitely noticed with the clear wings, the hummingbird moths, essentially, we see them pollinating a lot of wild bergamot and the common milkweed. So these tubular flowers, they both have the proboscis that can reach down, and so we see them pollinating those quite a bit.
I put together a video. If you want to learn a little bit more about the species pages and the project, we have a YouTube channel now where you can actually view this video, which is almost 35 minutes long, where I kind of walk you through some of the other details about the pages.
Towards the end now, we just wanted to highlight some of the species. We didn't keep great notes early on, but we have found so many different species that really had not been recorded in Indiana. We just got one today, actually, which was this Dahl's clearwing moth. And it was found by Chris Joel, actually in Porter County.
Chesterton's own.
Yeah, and yeah, right there in Chesterton. I don't know exactly where he was there, but we will look at other expert groups in the area, the moth photographers group, Bug Guide, Bold, Pomona, and just to, for verification in some cases. This one's actually currently waiting for verification, but most of these come back as accurate. And this moth has never been identified in Indiana based on about the 3 or 4 places that we look. Another one that was found, I think this was found in 2019. Oh, by the way, this clearwing is a wasp mimic. So if you saw that moth out on a trail, you may not even realize it was a moth. It looks like a wasp, but this is actually a very effective wasp mimic. It's exciting to have a new one. So this is the silvery blue, and we talked last night with another group about how unfortunate for the Karner blue it is that they seem to be extirpated. This species was also believed to be extirpated from the state. The last known area that it was seen was in the late 80s and early 90s, around that area, and was at the Hoosier Prairie. This species was just refound by Doug Seltzer in LaGrange County in Indiana, and he's gone back every year. He's found caterpillars and breeding populations of them right there in that area. So we've been able, through the project, to find this species back here in Indiana.
Nevada buck moth, Hemiluca nevadaensis is kind of a mystery. It's considered globally, it's a G3, globally imperiled, and these are moths that are found only in the Great Lakes region, and in Indiana we haven't seen them recently, up until last year when a few of them came in from I guess this was Kankakee Sands area, right?
Mm-hmm.
We also had one. Did we have one in Lake County also?
Lake County too, yep.
Beautiful moths. And then there's also invasive species. Well, excuse me, exotic species.
Yes,
like
so this is one that came from Japan, and it was Indiana's first record for this one as well, from Marion County, and it most likely is coming in on landscape plants. Okay, well, we're really excited to have everyone, just look for moths and butterflies. Cell phone technology makes captures, they don't have to, we've showed you some really stellar photos tonight, but that should not be any limitation. There's some people with really great photography and do a lot of neat things with the butterflies and moths to try to get amazing photos. Most of ours, photos come from people with the cell phone. All of my photos come from cell phone photos. And it can be very simple, just to turn on your porch light at night, and then go out there and take a look. A lot of people post almost nightly or in the morning with moths that they saw during the evening, or they just get up early and they check that porch light in the morning. So it's super easy to do, and easily facilitated through our group. But you can also email us if social media isn't the way that you want to do it.
And that, I think, concludes our presentation. So I'll turn off my screen share and be happy to take questions if there's time.
That was amazing. Amanda, thank you so much.
Yeah.
Oh my goodness. The work that you're doing is tremendous. I can't wait to send you pictures.
Oh, good.
That is so incredibly exciting. And it's almost like you've started your like brand new research project that could be housed at a university. I mean, oh my gosh, you are doing such tremendous work.
It's a, moths really
for
in
our-
particular, just easy, they're easy to find. Some are, but
Sure. Well, thank for all of the Lepidoptera lovers out there. I thank you for your incredible work. Oh, that was so exciting and so informative. In case you're tuning in a little late or are watching this later, we are recording this and are going to be posting it on our website. We'll send that information out for folks that want to tune in at another time or revisit the presentation like I will be doing. But I want to check in with Katie. Katie, do we have any questions in our Facebook chat for Steve or Amanda?
We do. Yeah. So Tyler asked, in reference to the physical differences between moths and butterflies you mentioned at the beginning of your presentation, are there any ideas as to why moths lack clubbed antennae?
Probably. Moths, moths have different types, many different types of antennae. So they have the thread-like antennae, which are called phyloform. And then they also have feathered antennae, which come in different forms as well. So if they're feathered on one side, they're called pectinate. If they're feathered on two sides, they're called bipectinate. And sometimes they're even feathered on four sides, which are called quadripectinate. We looked at the silk moths earlier, and one of the ways that we can tell the genders from some of the silk moths is the antennae on the females are typically much smaller than they are on the males. And the belief there is that the males have the larger, sometimes the females have bipectinate antennae, and the males of the same species will have quadripectinate antennae. And the idea behind that is the males need these larger, bushier antennas to go out searching for the females, which are not active, right? So like, unlike butterflies, which are out there visiting flowers, you might have a male and a female visiting, pollinating a flower at the same time, and they can find each other that way. In the case of many of these moths, and particularly the silk moths, the females are typically brown, dull, and or some form of camouflage, and they'll just go someplace where they're safe and stay put. And it's the male's job to go out and find them using the antennae to pick up the pheromones that the females are releasing. And so that's one possible explanation for it. And Amanda, do you have anything to
yeah,
add?
I think it almost certainly, and we talked about the olfactory kind of compounds on these antennae, and they're sensing food for the most part, but some are not, even eating as adults, but it's, reproduction really is the major drive for a lot of things. So I have a feeling that it's primarily the, searching for a mate, and in particular, males do have by and large, more ostentatious antennae, at least in the moths.
Excellent, thank you. I didn't see any other questions for Steve and Amanda. I do have one more for Desi, if you're willing. John asked if there are any residential areas that are a part of the test sites that you described.
Good question. We do not. Right now, we're really focusing on our efforts at the park. We have discussed the possibility of, with our restoration collaborations that we're doing in partnership with Save the Dunes and NIPSCO and Shirley Hines and other partners, we've discussed the possibility of adding some of those areas from Hobart Marsh northward to Miller Woods. I think it would be really interesting to add residential areas. I would love to do that. I think there's so much potential there with in terms of habitat connectivity and improving landscape scale connectivity for pollinators there, and it would be really interesting to look at that. It's, I think, probably mostly more of not an issue of interest, but rather of resources and capacity at this point. But something to think about and keep thinking about in the future, for sure.
thank you, Desi, Steve, and Amanda for such absolutely wonderful presentations, enriching content, scientific knowledge, and spreading the love of these not only fascinating and beautiful creatures, but essential creatures. Because remember, if it weren't for the pollinators, our plants wouldn't be able to complete their life cycle. The plants and the pollinators are linked, and we are part of that chain. We are part of that connection. So the more that we know, the more that we do, the more that we protect, the better we all will be. Did I just say that? The better we all will be.
I'm all for the bee puns.
Yeah, I love the bee puns.
Beautiful.
It's beautiful. So thank you all again. Thank you for tuning in, and please tune back in tomorrow. I'm going to share my screen one last time just to make a final plug for an extraordinary hour of pollinator art, photography, and culture tomorrow. And then if you want to learn about that landscape scale conservation work on Friday, don't forget to sign up to read your copy, to receive and then read your copy of our new Pollinator Garden Landscaping Guide. And we'll see you tomorrow. Thanks for tuning in.
Thank you.